Aquaporins are components of cell membranes used to transport water. In this lab, you will investigate whether aquaporins function passively through facilitated diffusion or actively with the input of energy by active transport. To examine this question, we will use a potato. This is a normal russet potato from the store. I just purchased this from the store. I'm going to then cut this russet potato into cylinders, equal-sized cylinders, 4 different equal-sized cylinders. I'm going to mask the cylinders, and then I'm going to put the cylinders into 4 different solutions of sodium chloride or salt. We have a 0 molar NaCl solution, a 0.2 molar NaCl solution, 0.4 molar NaCl solution, and 0.6 molar NaCl solution. The way that I made these NaCl solutions is that I took salt, the appropriate amount of salt, and just dissolved it in the same amount of water, 100 mLs of water. I then took the solutions and poured them into these test tubes. After I masked the potatoes, I'm going to take each cylinder, place each cylinder into a different solution, allow the cylinders to sit in those solutions for 30 minutes, then take the cylinders out, remask the cylinders at the end, and get the percent difference in mass. Before we run this experiment, I want you to think about the experimental design. And what I really want you to think about is how to control the possible variables that are going to be influencing the various factors. We have a potato. We're going to be cutting cylinders out of the potato. We have 4 different salt solutions. We want to think about controlling all the variables associated with those. We're going to be massing the solution. So I want you to think about all of the potential variables that could be influencing it and how we're going to control those with the experimental design. There are several variables that we need to consider controlling when we're doing this study. One important variable and relates to the sizes and the shapes of the potato pieces themselves. We could take a knife and we could cut the pieces into nice long rectangles that have the same size as close to we could by taking maybe a ruler and looking at the sides. No matter how we did that, then our potatoes wouldn't be exactly the same size. They'd be slightly off. Another method then of getting the same size pieces would be to use a cork borer. This cork borer then, if we use the same cork borer for all the pieces and cut the pieces at a specific size, their diameter and shape would be exactly the same. So we're going to use a cork borer when we go through and do this for all the pieces. Second variable we need to consider is how much solution needs to be placed inside these test tubes. In some cases, the amount of solution might be important, but more importantly, it's the surface area of the solution that contacts the potato cylinders themselves. So in this case, we need to make sure that our potato cylinders are just underneath the top of the solution so that we have full contact by all the solution with the potato cylinders. Another variable that you may not think about is when you take a potato out of each of these solutions, it has water on the outside. The water may hang differently, like if one drop of water drops off on one, that could amount to quite a bit of a difference mass-wise. So we need to make sure that we blot each potato. So we're going to take the potatoes out, blot them fully on Kim wipes, 1 round of blotting, then we're going to mask them before, and we'll blot them after to make sure that, that the water hanging there is the same. Now that we have all of our methods planned out, we're going to run the actual experiment. So I'm going to take this knife, I'll take the potato, I'm going to cut it straight across. We'll then measure out about 5.5 centimeters so that we can make sure each cylinder is the same length. Put those aside, take our cylinders and just put this straight down through there. Push this out. We have 1, 2, 3, 4, 4. So our cylinders are all ready to go. I'm going to make sure then they're exactly 5 centimeters. 1, 2, 3, 4. Now what I'm going to do is I'm going to blot the potatoes off. We'll grab a little whey paper here and we'll get this mask going. Okay, so we'll zoom in a little bit so you can see those masses themselves as we mask these guys. Okay, I'm going to tare it. I will now weigh it. You can actually get the weight yourself and record that in your manual. This is the 0 molar. I'll place it in the 0 molar. I'm going to blot and then put this on here. Let it sit. You can mask that one and record that one in your lab manual. I'll put it in the next in the 0.2 molar. Then going to blot the third one. We'll mask that one. You can record that in the manual under the 0.4 molar. And then going to blot the last one and mask it. You can mask that one and record it in your manual under the 0.6 molar. Okay, so now I've put all the potatoes now in these solutions. They're going to sit for 30 minutes and then we'll go in and mask them at the end. All right, 30 minutes have passed and so we're now ready to go in and mask our samples afterward. So I'm basically going to take each sample, place them on a different Kim wipe so that we can get the mass. Okay, this one, if we look at it, it's really hard and rigid. That's our 0 molar. There's our 0.2 molar. There's our 0.4 molar. And there's our 0.6 molar. Okay, so I'm going to take this one, blot it, and then I'll throw it over here on the scale and mass it so you can record that mass. We'll then take it off. That's our 0 molar. Here's our 0.2 molar. Okay, our 0.2 molar is a little bit flimsy. That 0 molar was really rigid. I shall put it on the scale, mass it, so you can get that 0.2 molar mass. Take that off, then we'll blot the 0.4 molar. The 0.4 molar is a lot softer as I can feel it and is more bendable. We'll put it on and mass it. And finally then we have our 0.6 molar. So we will blot that one dry and place it on and mass it.